apart from discovering hepcidin, you also discovered erythroferrone, the hormone that controls iron availability for red blood cell production. How was erythroferrone discovered?
We were looking for it deliberately. We knew that there was something made in the marrow that suppressed hepcidin. We also knew that if we took a mouse and bled it, hepcidin would come down. So what we did is we did an unbiased screen for what it is that the marrow was making after we bled the mouse. So we bled the mouse and we looked over the next 4 to 9 hours or so to see what turned on in the marrow in response to the bleed. And we specifically looked for secreted proteins, because whatever it was in the marrow had to be secreted into the bloodstream to act on the liver to modify hepcidin production. And we got very lucky. There was only one thing, one protein that fit this description. That is, it was made in between 4 and 9 hours after bleeding, and it was secreted. And this particular RNA or mRNA encoded a protein which at that time was called FAM132B. And that was a systematic name, but nobody really knew what, if anything, this protein was doing. And so we did a bunch of studies to show that, in fact, this transcript encoded a protein that modulated hepcidin. So we made mice that didn't make this protein. And in fact, they did not suppress hepcidin. After you bled them, they wouldn't suppress hepcidin. So that was the first indication, strong indication, that erythroferrone was required for this rapid suppression of hepcidin after a bleed. The second indication is that if we made erythroferrone and injected into mice, it would suppress hepcidin. If we made erythroferrone and put it on liver cells, it would also suppress the production of hepcidin by the liver cells. So in those 3 experiments, we essentially established that erythroferrone was maybe not the only, but the most important of the responses. The production of erythroferrone was the most important of the responses after bleeding that led to the suppression of hepcidin.
I actually remember when postdoc Leon Kautz came to your office to show the first results from mice that are lacking erythroferrone and show that they do not suppress hepcidin at all. And that was a real eureka moment.
Yes.
It involved some celebration.
Yes. By that time, we understood the system well enough that we knew what the results meant. It actually took several more years to do the opposite experiment, which is to make a mouse that overproduced erythroferrone. And that mouse ended up becoming iron overloaded and having higher than normal levels of iron in the body. So that was the last component of the proof that showed that erythroferrone can increase the iron absorption indirectly through hepcidin, and that erythroferrone is sufficient for creating this iron overload phenotype that is seen in certain diseases where erythroferrone is overproduced.
So we actually named erythroferrone, renamed it from FAM132B. How did that come about?
Well, we all gathered around the whiteboard and we wrote down names that would be descriptive of whatever this new hormone was doing. And my recollection is that there may have been 20 or so names on the whiteboard, and that we eventually selected erythroferrone because erythro means red, and ferrone means ferro, which is iron. So it connects red blood cells to iron. So it was erythroferrone, and "one" is a common ending that designates hormones. So it's a hormone that connects erythro to ferro.
And the nickname is ERFI, from erythroferrone.
Yes.
ERFI.
And most people prefer ERFI because it's much easier to say.